Hi, everybody. My name is Shervin Eskandari, and I'm a third-year undergraduate at the University of Georgia. My project consists of using deuterated pyruvate as a metabolic precursor of alanine to obtain high-resolution spectra of large proteins. Large proteins typically struggle with NMR resolution because they tend to tumble slower in solution than smaller proteins do. And we're going to attempt to resolve this issue by engineering an isotopically labeled form of alanine consisting of a 13CH2D group. And this form of alanine is going to give us sharp peaks in high resonance when we introduce it into a large recombinant protein. And we're going to use a methyl edited pulse sequence so we can quantify our peaks very well. So this background panel right here shows an experiment that was done similarly to what we're going to do here. However, we're going to attempt to bypass some of the limitations in this experiment. In this experiment, they used a 13C1 labeled glucose at the first carbon of glucose. And an issue with this is that glucose can go through many different metabolic pathways, as you can see. And if it does go through glycolysis and yield pyruvic acid, which is what we would want in this case, only half of the pyruvic acid compounds would be labeled. And this is because glucose actually gets split when it goes through glycolysis. So we can resolve this issue by starting with labeled pyruvic acid compounds so that all of our pyruvic acid is labeled. Another issue with this experiment was that they were using protonated compounds rather than deuterated compounds, so they didn't reap the benefits of deuteration when it comes to NMR. So this right here is the protein, and you can see the alanine residues represented as red sticks, and this is an HSQC spectra of the protein, with the horizontal axis being the C13 chemical shifts and the vertical axis being the proton chemical shifts. And these are all cross peaks of the alanine residues. So we created our BL21DE3 cell culture by starting our cells in LB growth medium, which is a nutrient-rich medium, and we incubated this for 16 hours, and then we inoculated our minimal salts media with kanamycin antibiotic, and we added our pyruvate in at about an absorbance value of 0.76, which was 3.5 hours into our incubation process. We then proceeded to take samples for 3 hours, once every hour, of supernatant and cell pellet samples. So this whole panel is devoted to the pyruvate methyl deuteration and the two different methods that we used. The first figure shows the first method that we used, which was our base catalyzed reaction. And this is the reaction mechanism that shows how pyruvate methyl deuteration can occur at the first carbon due to proton-deuteron exchange, yielding a fully deuterated pyruvate. This right here, figure 2, is our reaction monitoring. And this is an array of spectra. These spectra are all 8 minutes apart. And in the first one, we can see that we have a fully protonated CH3 group. And as you go up every spectra higher, you can see that the intensity of the peak decreases, the shift go to the right, and the splitting pattern changes. And these are all indications that deuteration is taking place. Figure 3 is the shift-based equilibrium side reaction of the alanine transamination catalytic mechanism. And this is the point where deuteration can occur via the use of the enzyme. And you can see that through this mechanism that proton-deuteron transfer can occur at the methyl carbon. Figure 4 shows two spectra, the top one being prior to the alanine aminotransferase enzyme, and the bottom one being 48 hours after the reaction has been completed. And in the bottom one, we can see that 3 species are present, the leftmost species being a CH3 group, the middle species being a CH2D group, and the far right species being a CHD2 group. Our BL21DE3 cell growth curve is shown here in figure 5. The horizontal axis is time, and the vertical axis are the absorbance values. We can see that the deuterated pyruvate was added in at 3.5 hours in, which is indicated by the red bullet point, and this did not affect our growth curve at all, which is a good thing for us. Figure 6 shows the alanine methyl group region of our cell supernatant at the first hour, and we can clearly see that something is here, which is a good sign for us. Although we can't quantify this peak because we don't have very good resonances and a sharp peak, this is a good point for us to build off of in the future. So through this study, we can conclude that the pyruvate methyl carbon, for our purpose, it was best deuterated through a base catalyzed reaction. However, we did notice that at higher pyruvate concentrations, a paradimer can form, a parapyruvate dimer can form. And we're going to try to bypass this in the future by using an acid catalyzed solution, which works with pyruvic acid rather than the more nucleophilic pyruvate compound. And we're also going to study the kinetics of the alanine transamination reaction in the future to try to maximize the concentration of the specific alanine isomer with a 13CH2D group. And we're going to express this isomer into a large recombinant protein and study it with a C13 methyl edited pulse sequence in the future and be able to quantify our peaks of large proteins. Thank you.